Okay, so welcome back to this next video in which we are discussing voltage-gated ion channels. Okay, right, so we're in the process of discussing voltage-gated sodium channels then, and we've seen that the main pore-forming portion of a voltage-gated sodium channel is made up by a single protein called the alpha subunit of a voltage-gated sodium channel, and you don't need anything other than the alpha subunit of a voltage-gated sodium channel to make a functional voltage-gated sodium channel. Okay, we will see later on how you can add accessory subunits on, but they're not necessary to have those. Okay, now we've seen that the structure of an alpha subunit of a voltage-gated sodium channel is very much so analogous to just having 4 alpha subunits of voltage-gated potassium channels joined together, okay, and these 4 separate portions, which are analogous to 4 separate alpha subunits of voltage-gated potassium channels, are called the 4 separate domains of the voltage-gated sodium channel. And we've seen that the way it then folds up makes the domains effectively equivalent to the individual subunits of a voltage-gated potassium channel. Okay, we've then discussed how the reason that the channel is usually non-conductive in the resting state is again because of the S6 helix, or rather the S6 helices from all 4 domains converging at the internal aspect of the pore and sealing the internal aspect of the pore and stopping sodium ions from being able to move through. Okay, and that's the so-called V-gate. Okay, so if you want to open the voltage-gated sodium channel, what you have to do is move the S6 helices outwards, and we've discussed that again, the mechanism for doing that is that when you get depolarization of the electrical potential difference across the cell membrane, that's going to allow the voltage sensing domains to move upwards because the S4 helices are all positively charged and will be feeling less of an attraction now to the intracellular compartment. Okay, and when the voltage sensing domains all move upwards, that causes a pulling on the S4, S5 linkers, which ends up pulling the S6 helices outwards. Okay, right, so what we now want to turn our attention onto is the permeation of sodium ions through the voltage-gated sodium channel. And of course, in this case, they're going to be moving from the extracellular fluid into the intracellular fluid, whereas in the case of the voltage-gated potassium channels, they were moving from the intracellular fluid to the extracellular fluid. Okay, and we want to talk about what is understood about how a voltage-gated sodium channel selects for sodium ions over other types of ions, okay, but it's less well understood how sodium channels select for sodium ions than it is for voltage-gated potassium channels selecting for potassium ions. Okay, right, so the first thing then that I want to discuss, if we're going to discuss permeation of sodium ions through the channel, is the hydration shell that sodium ions have. Okay, that's the first thing we discussed for potassium ions, and indeed when we were discussing it for potassium ions I showed you the hydration shell for sodium, but I just want to now remind you of that hydration shell for sodium. Okay, so sodium ions are a smaller ion then than potassium ions, basically, okay, and they can coordinate fewer water molecules around them than potassium ions were capable of coordinating. So here is our sodium ion, and now let me show you the water molecules surrounding them, and of course it will be the partially negatively charged oxygen atoms of the water molecules that will be pointing in towards the sodium ion, and the hydrogen atoms, which are partially positively charged, will be pointing away from the sodium ion. Okay, right, so what you're going to have in the case of sodium then is 6 water molecules surrounding the ion. Okay, so here's a ring of 4, and then you'll have 2 more, one above and one below, like so. Okay, so this then is the hydration shell for a sodium ion. Okay, and I might just connect all of the little bits up to make a geometric shape here. Okay, right, now basically if I now draw you a picture of what the open voltage-gated sodium channel looks like in cross-section, we will see how it's very analogous to voltage-gated potassium channels, i.e. the sodium ion can move with its full hydration shell on the internal aspect of the pore, but in the external aspect of the pore, where you have the selectivity filter, it's going to be slightly different. Okay, so let me now draw you a cross-section then of the pore of our voltage-gated sodium channel. Okay, so here we go. This is the extracellular side, this is the intracellular side. Okay, and here now is the pore down the middle. So we can see that once again, the internal pore here is much wider than the external pore here. Okay, the external pore is once again called the selectivity filter, and this is the narrowest portion of the pore. And again, it's going to be lined by those ascending portions of the P-loops of the four domains of the voltage-gated sodium channel. Okay, so these are the ascending portions of the P-loops of the four domains of the voltage-gated sodium channel. And this is what it will look like when it's in the open state. Of course, when it's not in the open state, the S6 helices will be occluding the internal pore. But in the open state, the S6 helices have been moved nicely outwards, and you have a nice wide internal pore. Okay, so we want then to see how sodium ions can move through this pore. So once they've got through the selectivity filter and they're in this internal pore, they will just have their full hydration shell of water molecules surrounding them, and they can just move through easily. Okay, however, let's discuss the selectivity filter then now. Okay, so the selectivity filter of voltage-gated sodium channels is actually wider than it was in the case of voltage-gated potassium channels. Okay, and this might come as a surprise initially. You might think, well, surely if we want to create a selectivity filter for sodium ions, we just need to make the binding sites smaller. Okay, so we need to make the selectivity filter narrower. Okay, because the problem with getting sodium through the selectivity filter of the voltage-gated potassium channels was that the sodium ions were too small, basically, and when they bound in those binding sites, they couldn't interact with the electronegative atoms at the corners of those binding sites very well because they were too far away from them, and that meant that they couldn't thermodynamically favourably give up their hydration shells to interact with these binding sites, basically. Okay, so you might think, well, just make a narrower selectivity filter, but that's not what happens. Instead, you make the selectivity filter wider, and the reason for this is that the sodium ion is not actually going to get rid of its full hydration shell in order to go through this selectivity filter here. In fact, it's only going to get rid of little bits of it and keep the rest, and it will interact with residues that make up the wall of the selectivity filter by getting rid of a few water molecules, but then where it's not interacting with residues that line the selectivity filter, it will maintain its hydration shell. So the sodium ion moving through this selectivity filter maintains part of its hydration shell, and the wider diameter of this selectivity filter in the case of the voltage-gated sodium channels is what then allows it to move through with part of its hydration shell intact. Okay, so let's now just explore this in a bit more detail and discuss selectivity, or at least what is understood of selectivity. Okay, right. So let me draw you then a little picture of the voltage-gated sodium channel selectivity filter. So again, the selectivity filter is lined by four ascending P-loops, basically. The four ascending P-loops from the four separate domains of the alpha subunit of the voltage-gated sodium channel, basically. Okay, and these are what I'm drawing here. Okay, so this is going to represent a square tube for our selectivity filter here. Okay, so what is this picture showing then? Each one of these lines, which I'm now going to colour in yellow, is representing an ascending p-loop of one of the domains of the alpha subunit of the voltage-gated sodium channel. Okay, so you could think of this as being the ascending p-loop for domain number 1. Okay, this is the ascending p-loop for domain number 2, this is the ascending p-loop for domain number 3, and this is the ascending p-loop of domain number 4. Okay, and together they make up the walls of this square tube through which the sodium ion can move, but not without its hydration shell. Okay, it will bring part of its hydration shell with it. Okay, so basically, all of these ascending p-loops are going to have carbonyl groups from the residues of those ascending p-loops facing into the pore. Okay, so analogous to in the case of the selectivity filter for voltage-gated potassium channels, the ascending p-loops are going to have carbonyl groups facing into the pore of the selectivity filter. Okay, and remember the oxygens of these carbonyl groups are very electronegative and therefore have this partial negative charge, and these carbonyl groups are going to be able to interact with sodium ions analogously to how the oxygens of water molecules would interact with the sodium ions. So what can happen basically is as the sodium ion comes through this selectivity filter, it can get rid of some of its water molecules. So for instance, it might get rid of these two here, and instead it might replace them with carbonyl groups from the side chains but rather from the residues of these ascending p-loops basically. Okay, and then it's just sort of going to slither through basically, or slide through by continuously forming and breaking new interactions where it's replacing water molecules with electronegative groups that are provided by the residues of these ascending p-loops that line the pore basically. Okay, right. However, you don't divide the selectivity filter up into those strict 4 binding sites anymore, the way we did for the selectivity filter of the voltage-gated potassium channel. Everything's much less strict, basically, in the case of the selectivity filter for voltage-gated sodium channels. In the case of the selectivity filter for voltage-gated potassium channels, there were those exact binding sites where the potassiums could bind. Here it's much more a case that there are just loads of little electronegative groups that the sodium ion could trade water molecules for to interact with, and it's just going to continue sort of busying its way down this selectivity filter, exchanging water molecules for electronegative groups that make up the walls of the selectivity filter, and then breaking those interactions, replacing them potentially with water, or forming new interactions with other groups further down basically. Okay, and that's how the sodium ion is going to move through. So I might just try and show this. So here is our sodium ion. Okay, so I'll colour it in red here, and it will be coming down with part of its hydration shell still intact. Okay, so some of the water molecules will be removed so that it can interact with the sides of the selectivity filter here, but it will still have some of the water molecules surrounding it basically, so it won't have got rid of its entire hydration shell. Okay, it will only be forming some interactions with electronegative groups on the sides of the selectivity filter basically, and then it will just break these and move down to a further portion, and that's how it can get through the selectivity filter. Okay, right, so that's permeation. That explains how sodium ions can actually get through the selectivity filter by interacting with these electronegative groups on the sides of the selectivity filter. And I should just say before we move on that it's not the case that there are exactly conserved residues in these ascending p-loops this time. Okay, so remember when we looked at the ascending p-loops of the alpha subunits of voltage-gated potassium channels, it was always that sequence of 5 amino acids in the ascending p-loop, the TVGYG motif, threonine, valine, glycine, tyrosine, glycine. Okay, it's not like that in the case of voltage-gated sodium channels ascending p-loops. Okay, there's not conserved residues, instead there's just carbonyl groups that are going to be facing into the pore of the selectivity filter to interact with the sodium ion as it moves through. Okay, however, having said that, there is one type of residue that is conserved always in the 4 ascending p loops from the 4 domains of an alpha subunit of a voltage-gated sodium channel, and indeed in all of the different 9 alpha subunits of voltage-gated sodium channels, and that's going to be involved in conferring selectivity. So we've now talked about permeation. Let's now talk about what is understood about selectivity. So firstly, let me tell you about this conserved ring that you find in, conserved type of residue, and it's not an exact residue, it's one or the other, okay, and it's found in all of the 4 ascending p loops of all of the 4 domains of an alpha subunit of a voltage-gated sodium channel, and that's the same no matter which alpha subunit you pick, whether you pick NAV 1.1, NAV 1.2, or all the way up to NAV 1.9. Okay, so let me tell you about this conserved portion. So basically what you have is a conserved residue that is either a glutamate residue, okay, and the 3-letter code for glutamate is GLU, or it's an aspartate residue, and the 3-letter code for aspartate is ASP. Okay, so glutamate or aspartate. Now let me firstly show you the structure of these 2 residues, okay, and then we can start to understand what this is going to mean and how this is going to have some effect on selectivity, okay, and I'm going to tell you what is known from experiment, but I'm not really going to be able to actually explain why what I'm about to tell you is true. Okay, so let me firstly show you then the structures of glutamate and aspartate residues. So we'll start off with a glutamate residue. Okay, so I'm drawing it as a residue again, so here is the amino group as though it's bound to the carboxylic acid group of the amino acid prior to it. Here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group. Okay, so that's the core amino acid structure here. And now what I'm going to do is show you the R group of a glutamate residue. Now, in a glutamate residue you have 2 methylene groups, so I'll just put CH2 for a methylene group and then bracket it and put 2, and that means repeat the methylene group twice. And then you have a carboxylic acid group. Now, under normal physiological pH, the carboxylic acid group ends up losing its proton to solution. Okay, so the definition of an acid molecule is a molecule that is capable of donating a proton to free solution. Okay, glutamic acid is the name for the molecule when it actually has the proton attached to it. Okay, so if you wanted me to draw you the structure of glutamic acid, this is not it. Okay, what I would have to do is have the proton there. Okay, so being very, very strict, you'd have to have the proton still attached to that oxygen, so you'd have an alcohol group there. Okay, instead, under normal physiological conditions, most of these residues don't exist as glutamic acid. Okay, so you don't really have that much glutamic acid because all of the glutamic acid molecules do donate their protons away into solution. Okay, they very rarely ever get them back, and instead they exist with a negative charge on this oxygen atom. And the strict name for this residue now is not glutamic acid, but glutamate. Okay, so glutamate is the conjugate base of glutamic acid. It's glutamic acid once it has lost its proton. Okay, and the reason people very rarely talk about glutamic acid is because most of the molecules exist as glutamate under normal physiological pH. Okay, so the key thing here to note is that glutamate has a negative charge, basically. Okay, it's a negatively charged ion. Okay, going to the other one, aspartate. Okay, the same thing applies for aspartate. Aspartic acid means the carboxylic acid with the proton attached to it, and aspartate is going to be the conjugate base of aspartic acid. So what is aspartic acid or aspartate? Well, it's very similar to glutamic acid and glutamate, okay? All you'd have to do is scratch that 2 there to turn this into aspartate here, okay? So glutamate, you have methylene group followed by another methylene group followed by this carboxylic acid group that's lost its proton and therefore has a negative charge here, okay? Aspartate, you would just have a single methylene group, not 2 of them, and then you'd have the carboxylic acid group that's lost its proton, okay? Aspartic acid is then the molecule which does actually still have its proton attached here and which only has 1 methylene group there, okay? Again, under normal physiological pH, you're not going to have that many aspartic acid molecules, okay, like you don't have that many glutamic acid molecules. So aspartate is the predominant form that these amino acid residues exist in inside cells. Okay, right, so basically every single one of these ascending p-loops from all 4 of the different domains of the alpha subunit of the voltage-gated sodium channel. So this one is from domain 1, this one's from domain 2, this one's from domain 3, this one's from domain 4. One of the residues that is at the top of the ascending p-loop will be either glutamate or aspartate. Okay, and that goes for all 4 domains basically, and it goes for all of the different types of alpha subunit of voltage-gated sodium channel. Okay, so let's say it's these residues here. These will either be glutamate or aspartate. So this one might be a glutamate, this one might be a glutamate, this one might be an aspartate, this one might be a glutamate. What's the significance of this? That you have a ring of negatively charged residues, because glutamate is negatively charged and so is aspartate, because they are these conjugate bases of the carboxylic acid molecules. They have lost their protons and they now have this negatively charged oxygen here. Okay, so you have a ring of negatively charged residues. This isn't partial negative charge here, this is true negative charge. Okay, so this is basically one of the things that somehow confers selectivity for sodium ions, basically, in these channels. You can understand why this is going to stop negatively charged ions moving through the selectivity filter. Okay, so a chloride anion now isn't going to move through here, simply because it's going to be repelled by this ring of negative charge. Okay, it's more difficult to understand why this might stop for instance, calcium ions or maybe potassium ions moving through. Well, the reality is that a lot of potassium ions actually do move through voltage-gated sodium channel selectivity filters. So voltage-gated sodium channels, their selectivity is not as good for sodium as voltage-gated potassium channel selectivity is for potassium, okay? Because this ring of negative charge, this is the answer, this is your selectivity, and we can see that that's not actually that rigorous at all, okay? It's going to stop negatively charged ions coming through, but there's nothing really to stop other positively charged ions coming through, and indeed these are very conductive for potassium. However, they're not so conductive for calcium as they are for sodium and potassium. So why is that? Well, it turns out that what actually happens is you can make this channel selective for calcium ions. You can make it into a calcium channel, basically, by engineering into this channel another ring of negative charge. So if you artificially, and I want to stress this is not how it actually is in nature, but if you artificially put another ring of negative charge in, so you give each one of these ascending p-loops another negatively charged residue, and you put them all in a ring like so, so maybe another aspartate or glutamate residue, and you then have 2 rings of negative charge. So I'll just draw these rings. So there's one, the normal one, and now there's this second one that we've put in. If you have those 2 rings, then suddenly that turns this into a calcium channel. So 1 ring, and it becomes a channel that is sort of selective for sodium, but lets in a lot of potassium. So I might just put sodium/potassium selective. Okay, it does prefer sodium over potassium, but you know, it does let through a lot of potassium. Okay, if you put 2 rings in, then suddenly that turns it into a calcium selective channel. Now we know calcium is a divalent cation rather than a monovalent cation, so obviously having two rings of negative charge is going to attract the calciums more, okay? So we will see later on that in fact the calcium channels, the voltage-gated calcium channels that we're going to come on to, they actually do have selectivity filters very analogous to the selectivity filters of voltage-gated sodium channels, but for the fact that you have two rings of negatively charged residues rather than one ring. Okay, so in fact when we were doing these experiments, we were actually turning voltage-gated sodium channels into voltage-gated calcium channels. Okay, right. So, I'll stress again that in the voltage-gated sodium channels you just have one ring of negatively charged residues, which are going to be glutamate or aspartate residues, and every single one of the ascending p-loops of all four domains of the alpha subunit of a voltage-gated sodium channel will have an aspartate or a glutamate residue at that level facing into the pore of the selectivity filter. And that's what gives us, to some extent, selectivity for sodium in these voltage-gated sodium channels. Okay, right, so that's now selectivity talked about, okay? We're now going to move on to the topic of inactivation, and then we'll talk about beta subunits of voltage-gated sodium channels. Okay, so first up then, we're going to talk about inactivation. Okay, right, so just like in voltage-gated potassium channels, voltage-gated sodium channels also inactivate. Okay, so when you depolarize the electrical potential difference across the cell membrane, we know that that will cause the voltage-gated sodium channel to open. But it's only transient, okay? It will only remain open generally for 1 to 2 milliseconds, even though you keep the electrical potential difference across the cell membrane depolarized beyond the threshold potential of around 40 millivolts. Okay, now why is that? Well, again, it's because there are inactivation gates. There are other things that you activate by depolarizing the electrical potential difference across the cell membrane, which are now going to try and close the pore, basically stop sodium ions from moving through. So basically, the reason that the channel becomes open for a second is because the opening of the V-gate in response to depolarization is quicker than the closing of these inactivation gates, which is also triggered by depolarization. And that's why you get this transient period where for 1 to 2 milliseconds you are open. Okay, so just like in voltage-gated potassium channels, there are two types of inactivation then, and we'll call these fast and slow type rather than N-type and C-type, although you can call them N-type and C-type. Okay, so we'll start off by discussing fast type inactivation. Now this is very analogous to fast type inactivation in voltage-gated potassium channels, in that you are going to get something occluding the intracellular entrance to the pore, basically, okay? And the thing which is going to occlude the intracellular entrance to the pore is that linker between domain number 3 and domain number 4, okay? That linker that I originally pointed out to you when we were looking at the structure of the alpha subunits of voltage-gated sodium channels. So let me just draw out the picture of the alpha subunit of a voltage-gated sodium channel once again. Okay, right, so let's start off by putting in domain number 1 here. So here is domain 1, then we have the linker from domain 1 to domain 2, here now is domain 2, then the linker from domain 2 to domain 3 here, then domain 3, okay, and then we have the linker from domain 3 to domain 4, which is the one that we're really interested in, and then here is domain 4. So here's the N terminus and here's the C terminus. So it's this portion that I've drawn this picture out for us to have a look at. This linker from domain 3 to domain 4 between the pore-forming domain of domain 3 and the voltage-sensing domain of domain 4. Okay, and it's this bit that is believed to be able to flip over, okay, into the intracellular aspect of the pore and occlude it, basically stop sodium ions from being able to continue moving there. Okay, and it's often compared to putting the lid over a can or something, closing off the pore by putting a lid over it. Okay, so it's often compared to a hinged lid, basically. Okay, now in this free-fall linker here, there is a sequence of 3 amino acids which is really important for the free-fall linker to be actually able to bind to the internal aspect of the pore and seal it off. Okay, and this is what's called the IFM motif. Okay, so again, in order to understand this, you need to be able to read single letter amino acid codes. So let me just go through these 3 different amino acids. So basically, this is a sequence of 3 amino acids that is found in the linker between domain 3 and domain 4 that seems to be incredibly involved in binding the free-fall linker to the internal aspect of the pore and therefore putting the lid over the internal aspect of the pore and stopping the movement of sodium ions into the cell through that pore. Okay, right, so what do these 3 letters mean then? Okay, so the first one, I, is for isoleucine, okay? I is the single letter code for the amino acid isoleucine, and I'll just show these structures out and I'll draw them out. So here we'll have the amino group then of the isoleucine residue, then we'll have the alpha carbon here with a hydrogen coming off it, and then here is the carboxylic acid group here. And then the R group of isoleucine consists of 3 carbons, like so, okay, and then you're going to have a methyl group coming off this first one here, okay, and then hydrogens coming off all these other 3 carbons here. So it's a 3 carbon side chain with a methyl group then coming off the first carbon of that side chain. Okay, right, so that's the R group then of this first amino acid, which is isoleucine. Then after the isoleucine, what you'll have is F. Now, what does F stand for? Okay, well, it stands for the amino acid phenylalanine. Now, phenylalanine couldn't be given the single letter code P because the single letter code P had gone to proline. Okay, so people deciding the single letter codes for amino acids said, what's the second best letter we can give for phenylalanine? Well, of course, phenylalanine sounds as though it begins with an F, so we'll give it F as its single letter code. Okay, right, so now let's put in the phenylalanine residue here. Here's the amino group here. Here's the alpha carbon and the carboxylic acid group, so that's all the core amino acid structure. And now let's put in the R group of phenylalanine. Okay, here is the methylene group. Okay, and then phenylalanine and then has a benzene ring. Okay, this is why it's called phenylalanine. The phenyl here means you've got a benzene ring attached onto alanine. Now, the amino acid alanine, its R group is literally just a methyl group, so this bit is the R group of the alanine effectively, and then you've stuck a benzene ring on the side, like so, and that's why it's called phenylalanine, because it's just alanine with a phenyl group stuck on the side. Okay, right, so that's phenylalanine here, the second amino acid in this IFM motif, and finally then, the third amino acid here is going to be a methionine residue. Okay, so here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, that's the core amino acid structure, and now we're going to have a methionine R group, and a methionine R group consists of an ethylene group, okay, so 2 methylene groups stuck together, then you have a sulfur atom, and then off the end of the sulfur atom you have a methyl group, and I'll just make sure this is still in view, yep. Okay, so here is the methyl group here, okay, and that is the structure of this final amino acid, which is methionine. Okay, so basically in the free-fall linker, you have this motif, this sequence of these 3 amino acids, isoleucine, phenylalanine, methionine, and that's present in all 9 of the different alpha subunits of voltage-gated sodium channels, and it seems to be really important for that free-fall linker to be able to bind to the underside of the pore and to block it basically like this hinged lid. Okay, right, so the free-fall linker only does that when you have depolarized the electrical potential difference across the cell membrane. Okay, so again, it is a gate that is responding to changes in the electrical potential difference across the cell membrane. So when you depolarize the electrical potential difference across the cell membrane, the free-fall linker is now going to cover the underside of the voltage-gated sodium channel alpha subunit pore. Okay, so it's going to cover up the intracellular opening of the pore of the alpha subunit of the voltage-gated sodium channel, and only when you then repolarize the electrical potential difference across the cell membrane back down to negative 65 millivolts will this come off, basically. Okay, so if I bring this diagram back here, it's again just like this. Okay, we have the closed state, which is the state where the V-gate is closed, and you are currently at an electrical potential difference of negative 65 millivolts, let's say. The V-gate is closed, so you've not got any sodium ions coming through. But the inactivation gate, for instance, this free-fall linker, will not be closed. It will be in the open state originally. Okay, so it won't be covering the intracellular opening of the pore. Okay, then you depolarize the electrical potential difference across the cell membrane, and the gate which is affected first, the gate which is affected quickest, is the V-gate. So the V-gate is opened in response to depolarization, and temporarily you go into this transient state, which is the open state, where the V-gate has just opened, okay, and the inactivation gate, this free-fall linker, has not had time yet to close, basically. Okay, and that process of going from the closed state to the open state is called activation. However, it's very short-lived, 1 to 2 milliseconds, because very quickly what's going to happen is the inactivation gate, the free-fall linker, is going to catch up. It's going to then close in response to the depolarization, and that process is called inactivation, and that takes us into this inactivated state where the V-gate is open but the inactivation gate is closed, and therefore you've got no sodium current coming through the pore. Okay, and both of those are triggered by depolarization. Then, if you want to now go back from the inactivated state to the closed state, you have to repolarize the electrical potential difference across the cell membrane, and this will cause the inactivation gate to reopen now that you've repolarized the electrical potential difference across the cell membrane, and that's called deinactivation, and you'll go transiently, incredibly transiently potentially, into the open state, okay, before then the V-gate closes again because of the depolarization, and that will, the repolarization, and that will then cause deactivation, this process of you going from the open state to the closed state because the V-gate is now closing in response to the repolarization. Okay, right, so I'll get rid of that flow diagram now. Okay, so that's fast inactivation, and that's the main one that now is responsible for the voltage-gated sodium channel then closing, even though the electrical potential difference across the cell membrane is still depolarized at the threshold potential. Okay, however, there is also, we think, a type of slow inactivation, a type of C-type inactivation, which, if you remove the free-fall linker, if, for instance, you remove the IFM motif, which is extremely important in the free-fall linker having an effect, the voltage-gated sodium channels will still inactivate, but it's much slower than they would if they did have the free-fall linker. Okay, and it's believed that, again, there is a mechanism for collapsing the selectivity filter so that it can no longer conduct sodium ions through it, basically. Okay. And again, that mechanism will be gated by depolarization of the electrical potential difference across the cell membrane. So depolarization will cause the collapse of the selectivity filter, albeit quite slowly, and then repolarization will cause the reopening of the selectivity filter. Okay, so again, it's another type of inactivation gate that's believed to be there in the case of voltage-gated sodium channels. Okay, right, so that's the topic of inactivation now covered. Let's now talk about these accessory subunits of voltage-gated sodium channels. So let's go over the page and talk about the beta subunits. Okay, right, so the first thing that I should say is that beta subunits are not necessary. You don't need to have them attached to the alpha subunit of the voltage-gated sodium channel to have a perfectly functional voltage-gated sodium channel. However, if you do attach beta subunits to your alpha subunit of your voltage-gated sodium channel, then they can modify the properties of your voltage-gated sodium channel. Specifically, they can modify what voltage it will actually open in response to. So they can modify the threshold potential, and they can also modify the rate at which the voltage-gated sodium channel will actually inactivate. So how quickly will you get the free-fall linker capping the internal opening of the pore and then closing the conductance, basically. Okay, right. So there are 4 different beta subunits of voltage-gated sodium channels. Okay, and these are called the NAV, standing for voltage-gated sodium channel, and then beta for beta subunit, and then there's the NAV beta 1, there's the NAV beta 2, there's the NAV beta 3, and then there's the NAV beta 4. So those are the names of the 4 separate beta subunits of voltage-gated sodium channels. Okay. Now let me draw out the structure then of all 4 of these NAV betas, okay? And again, this is just a broad structural overview of their membrane spanning topology. Okay, so basically they span the membrane a single time, and their amino terminus is going to be extracellularly, whilst their C terminus will be intracellularly. So here's the amino terminus of all of these different types of beta subunits of voltage-gated sodium channels, and here is the C terminus. So they have a single membrane spanning alpha helix. So that is the membrane spanning topology for all of these 4 different types of beta subunits of voltage-gated sodium channels. Okay, right. Now, it turns out you can attach up to 2 of these beta subunits of voltage-gated sodium channels onto your alpha subunit. Okay, but it's not exactly known where they can attach onto the alpha subunits. So what I'm going to do is show you a very crude picture, since we don't actually know exactly where they can attach. So the way I'm going to do this is I'm going to represent the entire great alpha subunit of the voltage-gated sodium channel once again just as a cylinder, like so. So there is our alpha subunit, and then I'm just going to have the 2 beta subunits that you can attach here on either side, basically. But I'm not going to be able to show you exactly where they actually attach on my more sophisticated picture of the alpha subunit of a voltage-gated sodium channel. Okay, so let's just colour this in here then. So here is my alpha subunit then, in red here. Okay, now let me tell you about the way that you can attach beta subunits onto the alpha subunit of a voltage-gated sodium channel. Okay, so basically it's not necessary that you attach any at all. You can attach just 1 if you want to, or you can attach 2. Okay, and the way that you can attach these is, there are rules to it, basically. Okay, so let's talk about this one here. So what you do is, when you're building a voltage-gated sodium channel, you have to now decide, do you want to attach beta subunits onto it? Okay, and let's now talk about the first beta subunit we're going to attach. So this first beta subunit you're going to attach can either be a beta 1 subunit, or it can be a beta 3 subunit. Okay, so on this side, this is representing the beta 1 or the beta 3 subunit. Okay, so if we're building a voltage-gated sodium channel, the first thing we'll do is decide which alpha subunit we're going to use. We've done that now. We're now choosing beta subunits. Okay, now, the first thing you then do with regards to beta subunits is you say, do I want to have a beta 1 or a beta 3 subunit? Or you could choose to have none of them. Okay, so you can either choose a beta 1 or a beta 3, or none of them, and you can put it in this site here. Okay, so you make your pick. Am I going to have none, or am I going to have beta 1 or beta 3? And basically you can choose one of these, and if you do choose to put one of them on, then it will be attached by non-covalent interactions. So the beta 1 or beta 3 subunit that you choose to attach onto the alpha subunit will be attached by non-covalent interactions. Okay, then the other beta subunit on the other side, this is either going to be beta 2 or beta 4 if you do decide to put it on. Okay, so at a different site you can either attach a beta 2 or a beta 4 subunit, or you can put on none there. You can choose to have that site unoccupied, basically. Okay, and when the beta 2 or beta 4 subunits bind to the alpha subunit, they do it through a covalent interaction. So this interaction over here is covalent. So let me go through those rules again. So you firstly decided upon your alpha subunit, okay, from NAV 1.1 to NAV 1.9. Then there are these two separate sites to which you can attach beta subunits. One of the sites can have either a beta 1 or a beta 3 subunit attached by non-covalent interactions. You have to choose only one. You can't attach both of them. You can't say I want a beta 1 and a beta 3. You can only put one of them onto that site. Okay? Or you can put none of them, of course. You can say I don't want anything in that site. Don't put anything on there. Okay, so you choose none, or a beta 1 or a beta 3, and you attach it to that site by non-covalent attachment. Then you've got another site on the alpha subunit of the voltage-gated sodium channel where you can attach either a beta 2 or a beta 4 subunit, and when you do that it will be by covalent interaction. Okay, so you can again opt out. You can say I don't want any, okay, or you can say I want beta 2 or beta 4 but not both, and you can't have two beta 2s attached or anything like that. Okay, you've got these two sites and that's it, where you can attach two beta subunits. One site is a non-covalent interaction and you can put either beta 1 or beta 3. The other is a covalent interaction and you can put either beta 2 or, beta 1 or beta 3 can go in this non-covalent site. You can put either beta 2 or beta 4 in this covalent site. I'm sorry if I've mixed that up. It's right on the piece of paper, basically. Okay, this site is non-covalent and it can have beta 1 or beta 3 attached. This site is the covalent site and you can either attach beta 2 or beta 4. Okay, or you can attach nothing at all if you want. Okay, right. So that then is the beta subunits. Now just once again dwell on what they actually do. So when you attach these beta subunits onto the alpha subunit of a voltage-gated sodium channel, then they can alter several things. So they can alter, for instance, the threshold potential for the opening of the voltage-gated potassium channel alpha subunit. Okay, that makes sense because they're going to be nearby the voltage sensing domains, so they might be able to interfere with how easily those voltage sensing domains can respond to depolarization of the electrical potential difference, and therefore make it more difficult for the voltage sensing domains to actually trigger the pore forming domains to open in response to depolarization, hence raising the threshold potential. So they can alter the threshold potential basically for opening. In addition, they can also affect how quickly inactivation occurs. So they can affect how quickly the free-fall linker will actually put that lid over the intracellular opening of the pore basically, and therefore affect how long the channel will be open for once it's actually been activated to open. Okay, right, so the final thing then that I want to say about voltage-gated sodium channels is to tell you about some very, very dangerous neurotoxins, very, very poisonous neurotoxins, which work by binding to the extracellular aspect of the alpha subunit of a voltage-gated sodium channel and blocking it. Okay, now why are these so dangerous? Well, they will do this all over neurons, basically, okay, and therefore they will stop neurons being able to fire action potentials because the voltage-gated sodium channels are utterly essential for action potentials, okay. If neurons don't fire action potentials, then you can no longer communicate. Your nervous system is going to break down, okay. You can no longer communicate to the diaphragm, for instance, to make it breathe. Also, of course, you've got voltage-gated sodium channels on your skeletal muscle cells, including the diaphragm, and they're responsible for action potentials in the skeletal muscle cells, which are essential if you want to actually make the muscle cell contract. So basically, these things can stop your breathing because of their blockade of voltage-gated sodium channels. Okay, so very, very dangerous toxins. Okay, so two really important toxins then are tetrodotoxin, and these are really important experimentally and also obviously medically to know about them. So tetrodotoxin, okay, which for short is abbreviated down to T for tetrodo and then TX for toxin. Okay, so tetrodotoxin. Now, tetrodotoxin is found within pufferfish. Okay, specifically it's found within the skin, the ovaries, the liver, and the intestines of pufferfish. Now, in certain parts of the world, including Japan, pufferfish is a delicacy. Okay, and therefore there is a huge danger here because you are eating something that is full, potentially, of this incredibly powerful neurotoxin. Okay, so pufferfish in Japan is only allowed to be prepared by chefs who have taken very rigorous courses in pufferfish anatomy, okay, and who are very competent at removing the bits which have tetrodotoxin within them. Okay. So that's a toxin that does occasionally, however, end up poisoning people because of slip-ups in the preparation of the pufferfish. Okay, so it's a very, very powerful neurotoxin because it blocks the extracellular aspect of voltage-gated sodium channels and hence blocks any sodium actually being able to move through the voltage-gated sodium channels, even if they're activated to open by the V-gate opening. Okay, they're not actually going to be able to conduct sodium ions. Okay, right. Another important example of a neurotoxin which works by doing the same thing, binding to the extracellular aspect of voltage-gated sodium channel alpha subunits and blocking them, is saxitoxin, which is often abbreviated down to STX. Okay, now saxitoxin is found within marine plankton, certain types of marine plankton. Okay, now, why is that a danger to humans then? Well, the problem is that certain shellfish, such as mussels, can eat these marine plankton which have the saxitoxin within them, and the saxitoxin builds up inside the shellfish. Okay, so the shellfish can accumulate more and more of this saxitoxin because they're eating more and more of the marine plankton. Okay. And then what happens, unfortunately, is the human will then eat the shellfish. And some of these shellfish, they can contain enough saxitoxin to kill huge numbers of people. And then if one human eats that entire shellfish, they don't stand a chance, basically. Okay, it causes very quick death. Okay, so again, a very powerful neurotoxin which works by blocking the extracellular aspect of the pore for voltage-gated sodium channels. So those are two important toxins to know about. They are very useful experimentally, and they are also important to know about clinically as well. Okay, right, because you do occasionally hear about toxicity due to these toxins. Okay, right. So in the next video then we'll move off voltage-gated sodium channels and we'll move on to voltage-gated calcium channels.